we are going to have a panel presented by CTI, and it is a panel all on innovation and research. We have some excellent panel members that are ready to come up and talk to you all. We have Matthew Huddleston, Executive Vice President and Chief Technology Officer for Enable Injections. He is an experienced medical device professional with more than 20 years of experience in startup environments with an emphasis on project management, design, development, manufacturing, regulatory, and intellectual property. He has issued several pending patents in the medical device field, including soft tissue and cardiovascular fixation devices and delivery instruments. And then next we have Dr. Christy G. Rothwell. She is a current partner at Fisher Broyles and former VP Legal Intellectual Property at Bexion. And lastly, we have William P. Swaney, who is the president of Expressions Manufacturing LLC and a renowned vector manufacturing expert. You guys are welcome to come on up. Alrighty, let's get started. This is a panel on innovation in research. You guys are welcome to start wherever you'd like. I have your presentation ready to go, so if you wanted to start, you can. Okay. I can do it for you if you like. Not a problem. All you're doing is clicking the space button to get through.
Which one?
Space button.
Awesome. Okay.
And that'll get you through. I can pull it up. Yeah, we're
It's
you
like
already sent
this,
it.
it's the third slide, and I can click on it.
Oh yeah, you just click on it and it should just go. Yep, it will play, and you should be good to go.
Awesome.
Alrighty. There we are.
Sorry. Thank you, everybody. Thank you, AHIS and CTI for having me today. Again, my name is Matthew Huddleston. I'm CTO and EVP at Enable Injections. Enable is forever in my mind will be a startup company, even though we're about 180 people in Cincinnati, Ohio. And we are solely focused on delivering large volume biologic drugs subcutaneously. And so I want to thank the Jasper team for the setup on this, because one of our applications is actually an oncology product, and it was just recently announced our collaboration with Sanofi on their isatuximab for multiple myeloma. But the thought here is rethinking drug delivery. And so with a class of biologics that have been developed for any treatment from autoimmune to oncology to rheumatoid to rare disease, a lot of these drugs are delivered IV. And this is inconvenient for the patient, it's inconvenient for the providers. And so rethinking drug delivery caused us to say, okay, there's got to be a better way. And so if you look at the opportunities now with respect to IV, there's syringe and needle, there's syringe driving pumps. I think what was mentioned before was, really COVID exacerbated the need for something different. And so Infuse was started in 2010, and I wish it was, for all of us who have developed technology, it's never a straight line. We actually started in 2010 thinking that we were going to make painless vaccine delivery devices. And I recognized very early that pain is so subjective, and I'm the biggest baby at Enable, right? And I've injected myself with plenty of products, and we started with sterile water. And it didn't matter how slow you delivered it, because that was our value propositions. We were going to deliver it really slow with really small needles. It didn't matter, it still hurt. And so we pivoted because there was a class of products called biologics that were being developed that were requiring larger volumes. You know, started out at 1 to 2. An auto-injector does a great job of doing that, but when you start talking about delivery volumes, and pharma thinks in milligrams, we think in mLs, we have to do the concentration difference, but you really start talking about volumes that are 5, 10, 20, even more mLs, and so there needs to be a solution. So we developed Infuse with, and we're seeing, you know, again, and I've got a video here, but we see improved efficiency and safety, and I'll hit on it in a minute with respect to whether the therapy is being delivered in the clinic or can that therapy be transitioned to the home. And so flexibility of in-clinic or at home, hidden needle technology, and then providing our partners with really market differentiation, because we're a business-to-business company. We develop our technology and we provide it to our partners, the pharma companies, and they distribute it to the patients, providing them the best differentiation for their product. So I'll go to the next slide. So I'm gonna thank you.
No worries.
And then you might want to help me keep on time too.
I got you.
I can talk forever.
I will keep you on track. Don't worry.
Okay.
There's your video.
So it's a short little video showing the value proposition. A lot easier to see this than listen to me talk. So that gives you a basic overview of the value proposition Enable brings, and relevant to why I'm here today talking about the Infuse in the AI informatics section is, besides the wearable technology that we've delivered, recognizing that we're going to start in the clinic and move to the home, we looked at this and said, okay, what do we need to cover, or what can we cover in that loss of information? Because when patients are face-to-face with their providers, right, they're able to make sure that they're getting the right treatment, the patient's actually doing it. It surprises me to this day that there are still adherence issues related to oncology treatments. And so the fact that you can help monitor with adherence, looking at compliance, when you start moving these patients to the home. I remember asking the nurses, I said, if today you had a patient in your office and tomorrow they were at home, what kind of information would you like? Then look at the patient and say, okay, if you were in the provider's care today, but now you can able to do this at home, what kind of information would you like? So it's about information. And so if you look at the traditional, and this is the traditional connected healthcare model, and we've talked about this for a long time. Again, the Jasper teams brought up the economic effect of being able to implement these kind of solutions and getting providers and healthcare systems to buy into it because it's more work. But it does provide for the better outcomes for patients. We believe that. This is the flow of how we would see information. And again, with respect to the delivery of drugs with our technology, we're looking at what additional information can we provide about the injection. And so we're able to, as a mechanical engineer, I was proud that we were able to develop the Infuse without any electronics. However, there's not a way today that I can mechanize Bluetooth and some of the other electronic pieces, so we did have to add electronics to this, but it was in a meaningful way. And so being able to provide things around the device and around the patient that then flows through the ecosystem, you have information, you can do data analytics and those things, and we're just researching that now. Again, the most important piece is what's the return on investment. So with respect to some of the things that we believe will provide value in return on investment, the obvious one is adherence and compliance, especially when you're moving as many patients that can be moved out of the clinic into the home. And so things from patient behavior, medication adherence, and this is not just for commercial, but these are also in clinical trial evaluations. We can also look at adverse events. That's really key, especially when you move some of these therapies to the home. You want to understand, is there any issues going on with the patient? Not only systematically, but also then locally at the injection sites, which is what we're particularly focused on. So with respect to injection site reactions, and then obviously all the ability to measure additional things, not only about the device, but about the patient, is what we're focused on, and we believe would be the next step in the evolve of the Infuse, which is really what we started day one, is making a better delivery experience, providing providers more access to the therapies for the patients in their home setting. So that is Infuse in a nutshell. Thank you.
We're going to hold all questions until the end of the panel session, but if you guys do have questions, you will be able to ask your individual panelists. Next, Dr. Rothwell, do you want to go?
Sure.
Okay.
I don't have any slides.
We'll just play his again.
It's all kinds of videos there, right?
I will just put up this slide in the back.
Okay. Yeah, I'm just winging it today.
Oh, I like winging it.
So I'm Christy Rothwell. I have a Ph.D. in biomedical science and a J.D., so my background is mostly in intellectual property. I'm kind of here in two roles. So I recently transitioned from Bexion to Fisher Broyles, which is a national law firm. I have to say, like, whatever I say here is my own opinion and not theirs. And then I started there in September. Previously, I was in-house at Bexion for about the last 5.5 years. So Bexion is a small local startup here in northern Kentucky. They have a biopharmaceutical product they're working on. It's in clinical stage. It is a lysosomal protein and a phospholipid. They've completed phase 1 trials in pediatrics and adult patients. It was shown to be safe, and hopefully in the next month, they'll be starting a phase 1B study in adult patients and metastatic colorectal cancer patients. They also have a phase 1 study going more towards a proof of concept related to CIPN. So they're looking at neuropathy as well as cancer at this point. I was brought in to help them with their strategies. So they licensed this technology out of Cincinnati Children's. And as it developed, Ray Takehiko, the founder, knew that IP was really important and they were trying desperately to fundraise. So I came in-house and helped them with their R&D efforts, the regulatory, and to put in place an IP strategy that would put them forward until they were hopefully on the market and through the FDA. So I think in general, just using them as an example, I think for researchers and startups in particular, it's really important to start that process early, talking to an intellectual property attorney or someone who can talk to you about your overall strategy and how it fits with the business. I know a lot of times it's a big struggle, right? How much do you disclose because you need to get investors and you're not quite ready for your patent application yet. So there's a great tug of war there, and it's much easier to work hand in hand from the beginning than it is to try to fix it later. So that was part of my job there, and something that I recommend to everybody who's starting out as a startup. It will make your life easier later. So I'm sort of familiar with the business side and the regulatory side. There's a lot of, for the pharmaceutical industry, there's a lot of market exclusivities available that can tie into your intellectual property strategy. As you get close to approval, those are always very important. And I would say one of the other pieces of your portfolio that you should look at is trade secrets. It's often an overlooked piece of intellectual property that can be very valuable. For example, in Bexion's case, they've spent a lot of time and energy, years and millions of dollars on their manufacturing process. They're not quite commercial scale yet, but for now, all of that is being held as trade secret. It's not quite ready for patenting, and you don't want to eat up part of your patent term if you don't need to. So that's also another piece of IP that can be very important to startups. So I think my advice to all of you, I'm assuming most of you are startups, is start early, thinking about it, put a strategy in place, and work closely with your counsel and business people to keep them aligned moving forward.
We are ready for you. I have anything?
I have slides, yeah, they should be in there. I sent them to Caitlin or Katrin yesterday.
You can start.
Afternoon, everybody. My name is William Swaney, and I am the president of Expression Manufacturing, LLC. We're a cell and gene therapy company located here in Westchester, Ohio. Apologies, we're having a little bit of a technical difficulty where my slides don't appear to be here. Just real briefly, the cell and gene therapy community is developing, did we get them?
Yep, we got them.
Perfect.
I will have you pulled up in a jiffy.
Thank you.
No problem.
Fantastic.
All right, where do we go? Sorry, guys. This one here?
Yeah.
Okay. I thought I had everybody.
Oh, this is great.
Sorry, everyone. Just want to make sure we have slides. Okay.
There we go.
There you are. And you just use the space bar.
The space bar? Okay. Apologies about that. So today I'd like to talk a little bit about an artificial intelligence-driven expression cassette optimization program that we use to enhance cell and gene therapy applications. But I also want to go ahead and recognize our two speakers because their talks were really pivotal for the same things that we would be doing. As a cell and gene therapy provider, we would be actually using and making some of the medicines that would best be used in Matthew's infuse device, and Christy's information about intellectual property and the protection of your manufacturing process and getting involved with those types of considerations early on. That's been foundational to our strategy all along as we've developed this. We are clinical stage as well, and we are located in Westchester, Ohio, and we opened last October. I'd like to take a moment to just thank the organizers for giving us an opportunity to share our thoughts on the use of artificial intelligence and machine learning in developing drug products today. From the very beginning, medical and healthcare delivery relied upon basic research and discovery and preclinical development. And this is best seen in this overview slide of the common drug product development pathway. At the culmination of the research and development phase, you would typically have enough information to be able to make an IND application, which is your entry point into human clinical trials. And that then kicks off with phase 1, phase 2, and phase 3 clinical development activities that I'm sure the people at Bexion know all about, as well as Richard or Matthew's team. In phase 1, you're basically looking at safety and maybe some efficacy for some things like cell and gene therapy products. Phase 2, you're starting to pivot into looking for efficacy, and then by phase 3, you're trying to establish pivotal studies that you will submit to the regulatory agencies along with your R&D and your manufacturing information, with the hope that you'll culminate with a new drug application or a biologic licensing application to go ahead and be able to begin commercial manufacturing of those products. The regulatory agencies would review that packet and then go ahead and after that, if it's you get approval, they would go ahead and expect you to begin post-market activities, additional follow-up and monitoring of your products. And those materials would slowly but surely enter into clinical use in the practice of medicine. And that process, although depicted here linearly, is probably more of a true closed circle where it goes back and forth. And so that begs one of the obvious questions is where's the intersectionality between artificial intelligence and drug product development? And the short answer is it's probably everywhere. It's across the entire spectrum. From basic research all the way up to the practice of medicine, today's technology enables us to look at things that we would previously been unable to look at. And expression therapeutics is really focusing using AI at the earlier part of this pathway. We're using an expression cassette optimization that we term eco-platform for short, to go ahead and try to engineer better cell and gene therapy products that have enhanced expression and therapeutic efficacy when they get into the clinic. By doing that at this phase, it enables us to go ahead and quickly translate from the basic research into the studies, as well as take along further candidates that we believe will be better and more likely to go on into clinical trial and be successful. Our expression cassette optimization program is extremely user-friendly and is contextually aware of, it's agnostic whether or not it's to the use of lentiviral vectors, mRNA, or AAV to deliver the sequences. We go ahead and put that into the program, and it creates RNA sculpting. It looks at RNA structures, delivery. It looks at codon usages in the target tissues, as well as the different indications where the disease will be done. That then gets put into our platform where bioinformatics and machine learning predict and optimize the best expression cassettes that we can take forward into development. So these improved candidates are then selected for further in vitro and in vivo POC studies right at that phase, the end of research and development and preclinical stage. And then the most promising cell and gene therapy candidates are then taken forward. This is what the user interface of our platform looked like. It offers unparalleled customization, and as we mentioned earlier, it really gives us a choice to choose which tissues and targets we want to express our gene product in. Are we looking to express in hematopoietic stem cells, hepatocyte cells? Would we be looking to go ahead and express in muscle? And again, it'll go ahead and it'll look as whether or not we're going to deliver that therapeutic gene using mRNA, AAV, or lentiviruses, and then optimize that. One of the things that the algorithm does for us very nicely is it takes a look at the mRNA that will be expressed upon viral gene delivery, and it actually then goes ahead and makes alterations in the primary sequence of the messenger RNA to improve the secondary and tertiary structure of the RNA, leading to enhanced protein expression and stability of the message. This is very similar to the same technology that's being used by Moderna and Pfizer Biotech during for the COVID vaccines as well. And this is really highlighted in this slide here. One of our primary targets that we focus on is a bleeding disorder, hemophilia A, where patients are missing factor VIII. And factor VIII is a very difficult protein to engineer. It's a very difficult protein to be delivered via gene therapy and viral vectors. And it's also, when you deliver it, it's very difficult to go ahead and get it to express in the target tissue where you want to express it. One of the factor VIII clinical trials at a different company right now looks on AAV factor VIII delivery in the liver. And as you can see right now, we took the factor VIII sequence, put it through our ECO platform and optimized it for delivery. And when we actually evaluated that in a in vitro model where we optimized that using liver optimized cassette platforms versus human optimized codon usages, what you can see is that we got about double the expression that you got by just making a better vector. We were very pleased when we went ahead and delivered that then in an in vivo model and put that in, and the very same construct that you can see on the right basically produced about 8 times more protein. And so the product that we would deliver then would be more therapeutic and more potent, enabling us possibly to use less product, which is advantageous in a scenario where you might have an immunogenic response because you gave more product. And so this is a very powerful tool that enables us to do better studies. We also use this technology, the AI platform, to go ahead and take a look at our 3rd generation lentiviral packaging plasmids. And what we were able to do there was we used the technology to increase the expression of the envelope and the Rev proteins, as well as use the platform to redesign the homologous, the overlapping regions of the plasmids, lowering the amount of homology between those backbones, thereby reducing the chance that there would be a homologous recombination of the plasmids during lentiviral manufacture, which would result in a replication-competent virus being produced. Also, importantly then, we took the transgene, which is the backbone that expresses the transgene, and we put structural motifs in there that would prevent translational read-through of open reading frames that were undesired or internal promoters. And what this does is it allows us to make safer products. Following that up, we wanted to go ahead and mention that from the time that a sponsor or our own products or provide us the sequence, we can integrate that into our lentiviral packaging system, produce scalable materials, very much like Christy was mentioning, research grade and then pilot grade, and eventually moving into a GMP manufacturing, then move to engineering scale up of that in about 1 or 2 months, and then eventual GMP manufacturing for entry into that clinical trial, like phase 1, phase 2 boundary that we depicted earlier in the drug product development. So that basically would enable people to go from ideal candidate into GMP clinical trial initiation in patients in the best case scenario in 1 year, which is really unprecedented because typically takes about maybe 18 months to 24 months to do the same type of thing. The really high point of all this technology is that the artificial intelligence is making better products, safer products, and it's iterative, it gets smarter as it learns and it goes ahead. And it really is offering us an opportunity to give physicians and those that practice medicine tools today to treat disorders like cancer and congenital disease that they normally wouldn't have had even 10 to 15, 20 years ago. With that, I'm going to go ahead and stop, and we can turn it over to the panel discussion. Thank you.
All right. Thank you all very much. Do we have any questions from the audience? Don't all jump at once. Everybody still waking up this morning? Okay. Come to the mic.
So, I have a question for the delivery, Mr. Huddleston, I think. There are 3 different ways that we deliver medications to patients, either IM, IV, or sub-Q. Does this address all of those, and are there only certain medications that can be delivered depending upon where they can be delivered to?
Thank you for the question. Now, we're solely focused on subcutaneous delivery. The type of drugs that would be applicable would be anything that could be infused in a bolus, as opposed to something that's metered, like insulin requires very fine delivery at a certain rate. So we are not that, but we could deliver anything that's safe to deliver subcutaneously.
So we're not talking about like IV antibiotics, and is all chemotherapy, are they all delivered sub-Q?
So there are types of chemotherapy that are delivered IV that cannot be delivered subcutaneously because of the cytotoxicity, but there's a class of immuno-oncologic drugs that can be delivered subcutaneously, either IV or subcutaneously, and are safe to be delivered under the skin.
Still a very exciting technology. Thank you for presenting.
Thank you for your questions.
Do we have anyone else before I release our panel? All right, thank you all so very much.